Dear students, after the initiation process, we will discussing now elongation in case of translation. So there are simply elongation. Elongation is the process which is the just simply addition of amino acid in a polypeptide chain. So in case of prokaryotes or in case of eukaryotes, elongation step is almost same. As we discussed in case of initiation, the sum of the parameters which are differentiating in between eukaryotic and prokaryotic translation, but elongation is the complete process which is similar, which is almost similar in case of prokaryotes and eukaryotes. Some of the elongation factors which are varying over here in prokaryotic and eukaryotic, but we are just negligible to that one and just starting the elongation of prokaryotic system. So there are three different steps involved in case of prokaryotic elongation. Number one is binding of amino acyl tRNA to the A site. How this binding of amino acyl tRNA to the A site occur, we'll discuss. Okay. So what is amino acyl tRNA? So I have already told you that amino acyl tRNA should be charged. So if it is charged with any amino acid, then only it can comes under the processing of elongation. So here we have a charged tRNA. The charged tRNA contains alanine amino acid, alanine having the sequence CGG, plus elongation factors were involved in the processing of elongation. So as in initiation factor in case of translation was there, so some of the elongation factor were working in the processing. So meanwhile we are discussing. So EF means elongation factor. TU is the specialized factor which works in elongation in case of step 1. So elongation factor TU with the help of GTP plus tRNA. Which tRNA? The tRNA which carrying the alanine is combined together to create the charged tRNA. Charged tRNA having alanine with the sequence CGG with GTP. Okay, this GTP is the occurring from the system only, elongation system only. So meanwhile you will see how GTP involved and how GTP we can utilize, and then further it will released by the system and utilized by the system. So we have charged tRNA as per the condition of elongation step 1, binding of amino acyl tRNA to the A site. So P site, A site and E site. So ribosome contain 3 sites. We in previous class we discussed already. So A, P and E. A is amino acyl, P is peptide-dyl and E is exit site. So in case of initiation, AUG is the methionine which is formulated in case of prokaryote, is already initiate the prokaryotic translation. Now the elongation step, and first step in elongation is binding of amino acyl tRNA which is charged in nature to the A site. So this charged tRNA which is combined together from the alanine with the tRNA plus EFTU with the high energy rich source GTP, it creates charged tRNA. Okay, this charged tRNA will come to the A site. This is the step 1. So P site already having the AUG which is formulated methionine, and now in case of elongation we have further alanine charged tRNA which will come to the A site. A site having the CGG sequence which is complementary to the CGG is the sequence of alanine which is the charged tRNA. Okay, so now the complex is having GTP, EFTU. EFTU is the elongation complex, GTP along with the assembly. Okay, so meanwhile what will happen after the elongation processes? So it utilizes the energy. So GTP will convert into the GDP plus EFTU. It will dissociate the assembly. This EFTU plus GTP will recombine with the help of another elongation factor, which is TS, and create the GTP. This GTP is further involved in the processing of charged tRNA. Clear? So how this GTP is utilized within the system, formed in the system, so there is the source of energy. Fine. So in case of second step, the second step of elongation is transfer of growing peptide from tRNA in P site to tRNA in the A site. What is the meaning? It means that the transfer of growing peptide means the P site is the site where the further processing is occurred, where the further processing of incoming amino acid will attach to the P site. So second step is the transfer of all the amino acid which is growing during the elongation, what transferred to the A site. Clear? How it will occur? So we have this E, P and A site. So what will happen? So on second step will tell, according to second step, all the amino acid which are present to the P site, they should transfer to the A site. So there is a formation of peptide bond. So there should be a bond in between amino acid, so that further elongation of polypeptide chain will be strong. Okay, so this bond will occur, and in the presence of EF, another elongation factor which is known as G, with the help of 1 GTP, this primarily bond will be strongest bond and transfer it to the A site. So our second step is also over here. What is the second step? Second step is the transfer of growing peptide. Transfer of growing peptide which was present in the P site should transfer to the A site. So from P it will transfer to the A. Clear? And there in between all the amino acid there should be a bond which is known as peptide bond. So this is the step 2. In further step 3 process, what is the third step in case of elongation? Third step is saying translocation. According to translocation, growing peptide tRNA which is present in the A site, it is second step, and according to second step, all the amino acid which are synthesized will transfer from the P to A with the bonding of peptide bond. Okay, so all the amino acid which are combined together with the help of peptide bond will again come to the A to P and P to A. So whatever the material is present in the A site will further transfer to the P site, and whatever the material which is present in the P site will come to the E. Clear? As the DNA synthesis, RNA synthesis and protein synthesis all in the direction of 5' to 3'. So this is the ribosomal unit which will move from 5' to 3'. So as their peptide bond will formed in between the newly synthesized amino acid and they are transferred to the A site. So another step is to vacate this A site for the further addition of new amino acid, okay, with the help of charged tRNA. So how it will go? According to the step 3, which is known as translocation. So according to translocation, all the material which is present in the A will transfer to the P, and all the material which will present to the P will come to the E. And E is the exit site. So exit site, what will the function of exit site? So there is no amino acid in the E site, exit site. So it will come out from the assembly. And we have newly synthesized polypeptide chain. And this is the step which are further repeated. So similarly, one by one, all the 3 steps will repeat out. So I repeat all the situations once again. This is the process of elongation in case of translation. It is the step 3. It contains 3 steps, 3 major steps. Number 1 is the binding of amino acid tRNA to the A site. Why to the A site? Because P is the site where methionine, which is formulated in case of prokaryote, is already present. So initiation already started. Okay, so initiation wala process almost ho chuka hai. So next further step is the elongation. So in elongation, according to elongation, the first step is the addition of amino acid tRNA to the A site. And how it will come? It will come with the help of some elongation factor like EFTU and GTP, which is act as an energy source. Okay, so it will comes like this only. The structure is similar to this one. So this is known as step 1. According to step 2, there should be a transfer of growing peptide from tRNA in P site to the tRNA in the A site. Means whatever the material which is present in the P is transferred to the A. So there should be a polypeptide chain synthesizing like this. Okay, so then in third step there should be a translocation. And what is the purpose of translocation? Translocation means exit of the uncharged tRNA, because all the amino acid which are present, which are comes under the elongation process, they are already present in the A site. And peptide bond is prepared in between the amino acid. So there should be vacancy of the A site, so that further amino acid will bind and they will come together in the A site, so that further protein synthesis will occur. Okay, so in translocation, whatever the material which is present in the A will transfer to the P, and whatever the material which is present in P will transfer to the E. And E is the site which contain uncharged tRNA, which having no use. So the assembly will discard this tRNA. Okay, or this tRNA will again utilized by some other purpose or in the protein synthesis as well. So from that way, the further processing will going on. Okay, further amino acid will come. It will bind to the A site. Then peptide bond formed in between the newly synthesized and mature amino acids, and then they will transfer to the A. Then further they transfer to the P, and further translocation is occurred. Then it is the process of 3 steps which is further going on until the stop codon will occur. So what is how the stop codon will stop the translation step? So I will just show you. There are some release factor 1 and release factor 2. There are 2 release factors were involved in case of prokaryote. Release factor 1 having UAA and UAG stop codon, whereas release factor 2 works on UAA and UGA. Okay, whereas in case of eukaryotes only one release factor will be stop the reaction. Okay, so these are having a shape like this one. You can say that this is the relief factor 1. So the relief factor 1 means it will bind to whether UAA or UAG. If it is RF2, then it will bind to UAA or UG. It will not bind to the UAG. So they are specific in case of prokaryotic system, but in case of eukaryote, only one type of releasing factors were involved, which are help in case of termination of translocation. translation. Okay, this is the translation and it is finished now. So students, you just read out the video with carefully, and whatever the doubt you have, you just mail me or WhatsApp to me, and keep on studying, and thank you.